Hello. Here we look at how proteins are purified from cells and then characterized. These studies begin with a cell fractionation scheme similar to the one we've seen for eukaryotic cells and tissues. Each successive spin separates progressively smaller organelles until you have all of the cell components in separate test tubes. At this point, proteins can be separated and purified from each cell fraction by first disrupting organelle membranes or disrupting protein-protein interactions. The goal is to first separate proteins from other kinds of molecules like DNA and RNA as shown here, and then to purify individual proteins from mixtures of proteins. How do we do that? Well, proteins differ in physical and chemical properties. First of all, they differ in mass or molecular weight. They also differ in size and shape. You may wonder, how is size or shape different from mass? Well, just imagine two polypeptides of about the same length. That means they'll be about the same mass, but imagine that one, when it folds into its three-dimensional shape, folds into a more dense, more compact conformation. That protein has a different shape than one that is folded more loosely. And there are techniques that will selectively separate molecules based on this size difference, and yet other techniques that will separate them based on differences in mass. Proteins vary widely in the degree to which their surfaces are positively or negatively charged, so we can take advantage of those charge differences to separate proteins as well. Charge, by the way, is also dependent on the pH of the water in which the protein is dissolved. Proteins also differ in solubility. In fact, larger proteins tend to be less soluble than smaller ones, and proteins tend to change their solubility characteristics if you increase the pH or decrease the pH, or likewise increase or decrease the salt concentration or ionic strength of the solution. We can make use of that phenomenon in a protein purification as well. And proteins are not all equally stable in their folded conformations. Some are more resistant to being denatured by heat or by change in pH or ionic strength. Alright, so now we have these cell fractions. Let's look at a few actual methods, some of many, for purifying proteins. Proteins in their natural or native state are often initially separated by some combination of size and charge. You can look up ion exchange chromatography on your own. That's a technique that actually separates proteins solely on the basis of difference in charge. Here we'll look at native gel electrophoresis. Charges at protein surfaces are conferred by the polar covalent groups on their side chains or R groups, or by acidic and basic side chains. Shown here is an acidic amino acid and a basic amino acid. The carboxyl group is the acidic group on the amino acid on the left. The amino group is the basic group of the side chain of the amino acid on the right. Well, at physiological pH, meaning pH around 7, a little higher than 7, the proton on the OH group of the amino acid on the left is lost, leaving this amino acid with a negative charge. Likewise, for the basic amino acid, the amino group can acquire a proton and become positively charged, again at physiological pH. So this is part of what accounts for the charge on the surface of proteins. A so-called flatbed starch or agarose gel electrophoresis setup is shown here, with a direct current or DC power supply connected to anodal and cathodal chambers, or sides of the gel if you will. Different protein samples containing a sample dye are placed in the sample wells. There they go. And the power is turned on. We would say a voltage or an electrical potential is applied across the gel. During electrophoresis, the sample dye, which is colored, can be seen moving towards the positive electrode, because the sample dye was chosen to be negatively charged at the pH of the electrophoresis buffer in this system. In fact, all the proteins are moving towards the anodal chamber as well. At the end of the run, when the sample dye nears the end of the gel, the power is switched off and the gel can be stained for all proteins generally or for specific proteins. Here we have the dark spots representing proteins that have been stained. In this kind of electrophoresis, the rate of movement of different proteins depends not only on charge differences, but also on the size differences between the proteins, since a semi-solid matrix composed of starch, in this case, provides frictional resistance against molecular movement. In other words, large proteins will move more slowly even if they happen to have more charge than some smaller proteins. A technique for separating proteins on differences in mass alone is SDS, that stands for sodium dodecyl sulfate, which you don't have to remember, or SDS, or denaturing polyacrylamide gel electrophoresis, sometimes referred to by its compound acronym SDS-PAGE. Proteins moving in SDS-PAGE are denatured, so that they differ from one another mainly in length, and that's a pretty close approximation to mass. Large-scale SDS-PAGE can be used to separate proteins, but in the end, the proteins remain denatured and may not refold into a biologically active molecule. Nevertheless, SDS-PAGE is a valuable tool for assessing the masses of polypeptides. And because quaternary structure is often based on weak interactions, it's also useful for analyzing whether an isolated protein, a protein you think is pure, is a single polypeptide or made up of two or more polypeptides. SDS-PAGE is typically run in a vertical gel apparatus like the one sketched here. Samples are applied at the top of the gel in sample wells, and when the power is turned on, the migration is in fact downward. But it is still set up to be towards the anodal chamber, towards the positive chamber. So negatively charged molecules are going to be moving down in this gel. Here's a cartoon of an SDS-PAGE gel after electrophoresis, in which otherwise invisible or colorless protein bands have been made visible by staining for general proteins. Whether the proteins were native, or denatured as in this case, they will be very highly resolved as thin bands compared to the larger, more poorly separated proteins on agarose gels. Now, if these samples were from different steps in a protein purification, the samples in lanes 2 and 3 might represent samples after 1 and then 2 purification steps. I hope you get the idea. In this PAGE or polyacrylamide gel from your textbook, a total native protein extract from cells was first electrophoresed in one direction, in this case left to right across the top of the gel. The gel was then soaked in SDS to denature the proteins that had separated, and then the whole gel was subjected to electrophoresis from top to bottom. This procedure is called two-dimensional electrophoresis and can separate many of the hundreds or even thousands of proteins present in cells. And you should get the impression that there are indeed many protein spots on this gel. If a single spot is cut out of the gel, and if it is in fact a pure protein, it can be partially digested with enzymes, and the results of that digestion subjected to mass spectroscopy as shown here. After a database search, you might actually find a previously identified protein whose sequence will allow you to isolate the gene that encoded your protein. This is a somewhat arcane example, by the way, of a genomics approach to understanding the functions of a specific protein. The last two techniques I want to mention involve determining the actual detailed structure of proteins. Once a protein is pure, it might be possible to crystallize it and subject the crystal to X-ray diffraction or X-ray crystallography. Well, making the crystal is the first step, and sometimes it's very tricky. There are actually many proteins that cannot be crystallized. But for those that can, the next step is to beam X-rays through the crystal and capture the pattern of X-ray diffraction on an X-ray film. Then the expert does the measurements and the math, with the help of a computer, of course, and comes up with a model for the protein, also built these days with a computer. And that is how we get these very pretty pictures of proteins in your textbook and elsewhere, of course. Another technique that can measure interatomic distances, like X-ray crystallography, is nuclear magnetic resonance, or NMR spectroscopy. When applied to a protein, you get an output such as shown on the left, a raw output on the left, and once again, the expert, aided with his or her computer, generates a model of the protein, in this case shown at the right. So I want to leave you with an understanding that there are actually many, many more techniques, especially for purifying proteins. Okay, I want to leave you with an understanding that there are actually many techniques for purifying and analyzing proteins, and this is just a small sample. So for now, we have to end this presentation.